Okay, we're going to try and bring it home. One more video. I'm going to try and get it all into one more video. Okay, so complementary bases. We touched on that a little bit, but here is the official slide. You have A with T, C with G, appearing with a pyrimidine. This, that they are held together, right, by hydrogen bonds. So we know just from basic chemistry that those are pretty weak bonds and that you can pull them apart. So keep that in mind because it'll come up later. Okay, now the next thing we need to know is how does DNA replicate, right? We know it's DNA, we know what it looks like. How does this thing replicate? So if this is your piece of DNA and you want to know like how does it replicate? The question is, does it look like this where so this is blue, this is the original. Is it like the original stays the original and you have this old piece of DNA and it just makes a brand new copy of the DNA? Right? So you have the old one and just a copy of the new one. Do you have one copy of the old and one copy of the new? So, you have one copy of the old one and one copy of the new one. Or is it like a mosaic, right? This is the dispersive theory, right? Which one of those is it? So essentially, how is the storage of information and sequence of bases actually replicated? Essentially, the model is conservative. The model is semi-conservative. They disproved conservative. They proved semi-conservative. So this is what it would look like if it was semi-conservative. This is what it would look like if it was dispersive. But we have semi-conservative. We will do some of these things in class, but just know it is a semi-conservative replication. Okay. Also, fun fact, Rosalind Franklin used this you can see the B form of DNA. So DNA has all kinds of replications based on the temperature and, you see it in all kinds of forms. We like to say, oh, it always looks like this because this is pretty and this is what Watson and Crick did. And it does. If you look at the B form, it looks like this. This is the B form. This is the B form. It looks more like photo 51, Rosalind's picture. But it actually is in all of these forms. It's in this like squishy A form, which looks like you just squished it. And it looks like this Z form, which is just like crazy. It is in a bunch of different conformations. So not all DNA looks as pretty as this. So Watson and Crick's DNA model is of the B form based on based on Rosalind Franklin's picture, right? This looks a lot like photo 51. And this is seen under aqueous, in water, low salt conditions. That's where you get the B form. And that's what they, because they stole our data. We also can see the A form, the Z form. It just is a little different, and you'll see that come up in things. Okay, RNA. Just like DNA, RNA has nucleotides linked with phosphate chains. The ribose sugar replaces the DNA. It replaces U with T. And of course, the exception is animal viruses sometimes have double-stranded RNA, which is just crazy. Viruses, super weird. So RNA can be single-stranded or double-stranded, but it's typically single-stranded. And I think in the quiz or several things it'll say, what's the difference between DNA and RNA? Typically, it has a ribose instead of a deoxyribose. It has uracil instead of thiamine, and it's single-stranded. That's typically the answer. 3 classes of RNA. Also, you have a messenger RNA. So this is the actual sequence of RNA that matches up with your DNA. You have ribosomal RNA, and that's like the big sandwich thing that comes down and actually performs the translation. And then you have transfer RNA, and we'll look at that. It's just the 3 forms that actually get you from DNA to amino acids with the ribosomal and the messenger, and the T, of course. This originated as complementary copies of the sequence of DNA. So the point of RNA is to get you from DNA to amino acids, and the intermediary there is RNA. That's what gets that done, the 3 forms. Oh, so again, you have ribosomal RNA. This is the structural component for protein synthesis. It's like a giant kind of hamburger thing that comes out. We'll watch a video. The messenger RNA is the actual genetic information. This is the sequence of RNA that matches up. And the transfer RNA actually carries the amino acids to the ribosome. It's the little thing that holds the amino acid on it. We will see it when we get to that chapter. There's also some other weird, of course there's weird RNA because there's RNA and everything that's weird. There's telomerase RNA and RNA primers. We will put primers in when we do PCR. There will be RNA primers. There's small nuclear RNAs and those process messenger RNA. There's antisense RNA. There's microRNA. I mean, there's just a bunch of these things involved in gene regulation. Okay, so let's talk really quickly about analytical techniques in DNA, and we will talk about this a lot when we get to transgenic organisms. This is just sort of a flyby overview of genetic things that are done analytically to look at DNA. So UV light, hybridization, fluorescence, and electrophoresis. So one of the hyperchromic shifts actually just subject it to heat. So once you get up into a certain temperature, it denatures or separates. This is real important in PCR. You're going to see the DNA separate. It's just an unwinding, and it's essentially when you heat it to a certain, just like things get denatured, right? An egg denatures, the protein gets denatured, the DNA gets denatured when you heat it. During this unwinding, the viscosity of the nucleic acid decreases, and it actually absorbs, its ability to absorb UV light also increases. A hyperchromic shift during DNA denaturization is used to determine the melting temperature, and the melting temperature is a method for estimating base composition of DNA. So that's how we know how many bases it has. This is just one way in which we do that, to look at for example, melting profile shows the increased UV absorption versus temperature for two DNA molecules with different CG contents. The molecule with the melting point of 83 degrees, that's this guy right here, has a greater CG content than the molecule here. Okay, molecular hybridization. This is separating just this part, right? We're separating this part of the strand and of the DNA, and we're going to put a little DNA profile there. We're going to put a little piece of what we want in here and get it annealed with the DNA. Again, this is going to come up when we talk about PCR. It will hybridize, and these probes, they can be fluorescent, they can be UV, they can be just a gene that knocks out something or does something else. FISH actually is when we use that and we put a fluorescent probe. So this is part of how we do the big sequencing, right? When we sequence a whole genome, a lot of that comes from fluorescent probes. So we have probes that only attach to the uracil, that only attach to the thymine, right? And then you can see, looking at that, you can see which is which. These are just probes that attach to look for hybridization. They only specific chromosome areas, so you can see where things are. So this is just a FISH, which is just a hybridization of chromosomes. Last but not least, we are going to make it. We have electrophoresis. We will be doing some electrophoresis if you're in forensics lab or if you took genetic lab. We will look at gel electrophoresis when we get into more DNA technology, but essentially it separates it out by size, right? And you have this completed gel and you can match them up, and this is how we used to do gene sequencing, and it took forever. And now we can do what it took me, 3 years to do like this. We can do it in, like a week. So again, it's agarose gel and it separates it out by size. The smaller the piece, right? If you look here, the smaller the piece, the further it can move. Does that make sense? It's like a bunch of matrices, like the agarose forms these little components that's hard to get through. And so the smaller ones can just go further. And you have a cathode, a negative and a positive, and it moves this way. And these guys are smaller and these guys are bigger. The fat ones just can't make it through because they're too big to fit through the little holes of the gel. Okay, and that is it, friends. We made it. Okay, I will see you Wednesday. Bye.